I've got another name up here who's not presenting today. And I inherited a lot of these slides from her. I modified many of them and added some more. But it's important to note that what you're kind of experiencing today and all that stuff is not something that got invented, yesterday. The agricultural nutrient management in Maryland has been developed over the course of 30 years plus. So there are a lot of people that have put tons of work into this, and she is the lady who ran the University of Maryland Agricultural Nutrient Management Program before she retired. It's my privilege to do so now and to get to work with fabulous people like Emily and Brian, who have done an amazing job today. So it's great to be here. All right, so we want to talk about soil fertility, and usually that means nutrients, right? What do we add? We want to grow stuff. It's important to keep in mind before we talk about nutrients and what we mean by that, it's just soil fertility in that small sense, to keep in mind that if we really are talking about growing plants, it's a lot more than just the amount of nutrients that we have, right? It's really this phenomenon that emerges from all the different states that the soil has. And do we have a situation that's basically healthy, right? Do we lack disease pressure? Is our soil, capable of helping plants to stand up? Is it too hot? Is there enough oxygen for plant roots? Plant roots do not get oxygen from the air. Plant roots get oxygen from within the soil itself, from within the soil porosity, right? That 50% of the soil that is in the ideal soil that is just the pore space, that's the place that plant roots are breathing from. And if a plant root can't breathe, it can't do work. So it can't take up water, and it can't take up nutrients. All right. So if you want your plant roots to be functioning, you got to have oxygen in the soil, and to have oxygen in the soil, you've got to have enough porosity, et cetera, et cetera. So we're here to talk about this, but really having an environment where it can support growth is about a whole lot more. So we've really got three main questions to answer today. Why are some soils better at supplying nutrients than others? How do soils lose their nutrient content over time? How can you tell? And what can we do to maintain the fertility that a soil has? And of course, there's another question here too. What could we do to actually increase the fertility of the soil, right? We're not going to talk about that latter one because that's one of these kind of emergent things of soil quality and some of the stuff that Brian was talking about, cover cropping, various ways to try to add more organic matter, to add more porosity, to keep that soil from getting compacted. All right? So we're talking about this, but there are bigger questions to answer and kind of bigger goals that anybody who wants to really take care of their dirt is going to want to be thinking about. All right, so let's talk about what defines a nutrient. All right. Well, a nutrient is required to complete the life cycle of a plant. And nutrients are not generated by the plants themselves. They come from outside them. Right? That's what the word exogenous means. They have to come from outside. They've got to be supplied by the environment somehow. Plants don't generate them. Plants generate vitamins, but they don't generate nutrients. All right? And then there's this kind of a basic model for the way a lot of folks think about nutrients in plants. Think of a barrel that's got these various staves, these parts that make up the barrel. And you can think of the barrel where each stave corresponds to a different plant nutrient. And whichever nutrient you're lowest in relative to what the needs of the plant are, that's the nutrient that is called the limiting nutrient. It's the one that's going to really keep you from getting where you need to go until you can kind of add more of it, add more of that stave, and then you're going to get a leak somewhere else, right? Then you need to add that nutrient, et cetera, et cetera. So this is a model, and it probably works in a lot of cases as far as we can tell. But keep in mind it's only a model, but it's the Liebig-Spregnell law of the minimum. All right. Now, Emily's been talking to you about complexity of what's going on in the soil. And of course, what plants do itself is equally complex. So chances are this Liebig-Spregnell law of the minimum is indicative, but does everything always work this way? Probably. Probably not, all right? But it's good to be aware of this concept, okay? Because to first approximation, it works, all right? But does it describe every situation exactly? Probably not. So here we go, plant essential nutrients, all right? These are the things that all plants have to have. Some plants need something else, like grasses like to have some silicon, all right? When you're kind of running your fingers through a grass and you do it the wrong way and you get a cut, right? Those are little silicon crystals that are cutting you. All right, so some plants want some other stuff, but here we go. The plants have to have carbon. They get that from the air, right? Pulling it out of the air. They've got to have oxygen. They've got to have water. And then they've got to have these other nutrients. The nitrogen, phosphorus, potassium, the primary macronutrients. Then we've got these secondary macronutrients, calcium, magnesium, sulfur. These things are definitely on the test. What are the primary nutrients, right? The primary macronutrients, know it. Nitrogen, phosphorus, potassium. What are the secondary macronutrients? Know that. Calcium, magnesium, sulfur, all right? And then we've got the micronutrients. Macronutrients means, obviously, the plant wants more of those. Micronutrients, doesn't need as much, but does it still need them? It still needs them. They've got to be there. Where you will have a problem if you don't have enough of those things. And what's interesting is notice how they were all charged. Look at these things are all charged, right? Maybe there's a form of boron that's not, right? H3BO4. But all these things have a charge. One of the ways that these things are going to be retained in soil is by a charge in the soil that's going to retain them, a negative charge. The neat thing is a lot of clay particles come with the negative charge associated with them. It's just how clays are built. When clays are formed in the soil, as they form, there's this negative charge that is built into them. And that negative charge is going to hold a lot of these, certainly these positive ions. A lot of organic matter has a positive charge to it. A lot of negative too, and so that's going to hold some of these negative things. So charge in the soil is very important, especially when it comes to retaining minerals. So that's a lot, right? But here's the good news. There's a useful mnemonic, all right? And you've seen, if you've looked at some of the captions for some of these pictures, you've seen this name before a while. He's written the definitive soil textbook used around the world. It's now in its 15th edition, and we are so lucky he is here in Maryland, right? He's had his career in Maryland. Like the person who wrote the definitive soils textbook could have lived anywhere. But where does he actually live? All this, so many examples from his book are from around here, the Mid-Atlantic. We're so blessed. So he's got this great mnemonic to remember all these things, and you should do that. And it's fun. CB Hopkins Cafe Company. Closed Monday morning and night. CU Zoom, the management. Now, where are all of these nutrients embedded in this mnemonic? What about the C? What does that stand for?
Calcium. Carbon.
Let's go with carbon.
Carbon.
I think the calcium's going to show up another way. How about the B? Boron. How about H? hydrogen. Tell me about the rest of Hopkins. Oxygen, phosphorus, potassium, nitrogen, sulfur. I love it. Tell me about cafe. Calcium and iron. Calcium was coming, right? Calcium and iron. What's the company? Cobalt. I'm loving it. How about closed? Chlorine. Monday. Molybdenum. How about, interesting, morning?
Oh, is it Mn together, manganese?
Manganese. Nice. Mn. So that morning, think of the Mn, manganese. Night. Nickel. Ooh, this is funny. Cu Zun. Cu Zun. What's that?
Selenium? No,
Selenium is not essential. Maybe there's some plants that like it, but they don't need it. But plants universally don't need it. Cu. Copper. Cu. Copper. That's it. How about Zun? Zinc. All right. And then the management. Magnesium. Well done. Okay. All right. So this makes it fun. All right. We got that. Okay. Now, what do plants do with these nutrients? All right. Well, there's nitrogen in every single molecule of chlorophyll. We're going to see this in a second. It's fascinating. Phosphorus is important because ATP, this molecule that helps to transfer energy around in cells, like that's how energy gets transferred, is through ATP. And what does that TP stand for? Triphosphate. So if you're going to have energy transfer occur in a cell, it's going to happen because of phosphate. And also DNA has this kind of phosphorus backbone. You can't have DNA without phosphorus, right? Pretty important. Okay. As plant roots suck up ions of a particular charge, the whole thing has to remain neutral the whole time. And so sometimes plants will exude an ion back out in order to maintain a balance. Sometimes they'll exude out a hydrogen, sometimes they'll exude out a potassium, sometimes it'll be a chlorine, right? Okay. Got to have magnesium to have chlorophyll. The magnesium's in the center. It's surrounded by nitrogens. We're going to see that in a second. It's pretty cool. All the other essential elements like magnesium, they play some indispensable role in the workings of cells. Each one is going to be a little bit different. But oftentimes these metals, magnesium, calcium, iron, nickel, they are grabbed by some organic grouping, carbons and oxygens and hydrogens, have grabbed them in a protein in just a particular way, with a particular shape, with that metal atom in its charge in just the right place that allows that molecule to get something done that can't be done any other way. All right? And it's going to be as complex as all the things that cells have to do. And you don't have to worry about that complexity. You just have to know that they're essential. All right? This is so cool. Look at this. On the left is hemoglobin. Hemoglobin does a pretty important job. Try breathing right now, right? What's that hemoglobin doing? It is the iron in the center is grabbing oxygen atoms and taking those oxygen atoms someplace else inside your body where those oxygens get dropped off to a cell so that your cell can work, okay? So that your cell can can respire and get some energy out of it and do a little bit of work to keep going until the next oxygen atom comes. Now, over on the right-hand side is something completely foreign to you and me, right? Chlorophyll. But golly, it looks pretty much the same, doesn't it? Now, what's the difference? There's some little details here on the bottom that are different, right? Some of the bonds of the nitrogens might be a little different.
The center is different.
The center is different, right? In the center we got an iron for us, in the center we've got a magnesium for plants. And what does that thing do? I mean, it's kind of cool that this the hemoglobin is capable of grabbing an oxygen and ferrying the oxygen around to drop it off someplace. But oh my goodness, what's that magnesium doing? It's doing something that is truly stupendous. It's taking energy in one form and transforming it into a completely different form, right? Which is knock your socks off, even more amazing. So this is an example how I was telling you that we've got, what are these metal atoms doing, making them essential? Well, they're being grabbed by these organic proteins, right? In just the right way, so that with their charge, they can do just the right thing, whatever that might be. All right? And maybe it's not a surprise that we're something like 60% banana, or something like that, right? Maybe it's not a surprise. Hug your banana today. All right, now, why are some soils better at supplying nutrients than others? Okay? Well, all things being equal, why is it that some soils are more fertile than others? And here's the beginning of the answer. It's that some soils contain more nutrients anyway. It's the beginning of the answer. Also, some soils hold on to nutrients better than others. So some have more nutrients to begin with anyway, just because out of how they formed. Others have the right stuff to hold more nutrients than other soils do. Excuse me. So why do some soils inherently contain more nutrients? Well, some soils developed from geologies that were more enriched with nutrients in the first place. All right? Now, so here we've got a bunch of minerals soils can be made out of. Here's their formulas. Keep in mind our mnemonic, right? CB Hopkins Cafe Company, closed Monday morning and night. See you Zoom the management. How many of those suckers are here in a soil that forms mostly out of quartz?
2.
If silicon even counted, right? But silicon doesn't even count. So if you're forming a soil mostly out of quartz, how fertile is it going to be normally, would you expect it to be?
Not so fertile.
No, you'd have to do some incredible management. Get a bunch of organic matter in there, right? In order to make that thing fertile, because it doesn't start off that way. Situation gets a little bit nicer when you start with olivine, right? You got 2 of them now. Okay. How about with kaolinite? How's that situation? Not so good, right? Aluminum is actually, you get too much aluminum. Well, there's tons of aluminum in all soils, tons of it. It becomes a problem only when the pH gets too low and that aluminum starts to be able to get dissolved into the soil water. If the pH is high enough, that doesn't happen and there ain't no problem. But if there's some aluminum that's dissolved in the soil water, enough of it, you actually can develop a soil that's toxic, especially for sensitive plants. But you could have a soil that's made out of kaolinite and would not be toxic to plants. Just keep that pH up. But it might have formed out of kaolinite, but there's some trace minerals that were there before that were too that were not kaolinite, right? Or that you've done some amazing management over time, got some organic matter in there, and stuffed it a little. All right. Situation a little bit better with muscovite, not so much better. Okay, montmorillonite, a little bit better. Limestone, okay, a little bit better. Here's the neat thing, is that no soil forms purely, typically, out of just one of these. All right. The parent material of soils is always going to be a mixture. It's usually a mixture of textures, right? As the rock or the sediment that got put down usually is a mixture of textures. There's some examples of that is not true for like beach sand. But it's usually a mixture of textures and it's usually a mixture of mineralogies. So you're never just starting out with one of these, all right? But you might be starting off with a predominance in one of these. Generally, the more you got, the better chance you're going to have of having starting off with a parent material that is more fertile. Okay, so some soils are richer because they just inherited more from the very beginning. Why do some soils hold more nutrients? Because soils differ in organic matter content. Organic matter has these charges that will hold these minerals like we saw from that slide, that table that was before the mnemonic, okay? And then soils differ in the relative amount of sand, silt, and clay they have. Emily told you earlier that one of those components, sand, silt, and clay, is better than the others at holding some of those mineral nutrients. Which one of the three, sand, silt, or clay, is the part that's better than the others? The clay. And why is that?
It's negatively charged because of high surface area.
I love it. Both answers at once, okay? We've got a high surface area, so there's just more to stick to, right? And as clays form, there's this negative charge that simply is created as they form. All right, we won't go into the details on why that is. That's going to be in Dr. Weil's textbook. But it's incredibly cool. All right? So organic matter has charge, and it's going to hold those minerals that themselves are charged, these essential plant nutrients. And then we've got sand, silt, and clay. The clay part is the part with lots of surface area, and itself is negative, so it's going to grab a lot of those positives. All right, and then there's different types of clays. Do you think all clays are equally good at holding, having a negative charge? No, some clays have a lot more negative charge than others. All right, so it's not just the presence of clay; it's what type of clay. All right, pH is also a factor. Remember what pH is. pH is, well, we're going to talk about pH, but here's the thing, and you already know this anyway. pH, what you're measuring with that is you're measuring the amount of dissolved hydrogen ions, okay? Dissolved hydrogen ions. And hydrogens are they're easy. They're a single positive charge. If you've got a lot of those around, right? So in other words, you've got a low pH, lots of acidity. That means a lot of these hydrogens around, and all these little positive charges around. Well, what are they doing? Chances are the negative sites that you have are going to be saturated with a lot of these little hydrogens, right? Because there's just so abundant, and they're kind of fighting to be next to those negative charges. And if you got a lot of hydrogens around, well, chances are, just statistically speaking, they're going to take up a lot of those spots. So if you've got a acidic soil, a lot of the spots that you'd like to have available to hold a mineral have been taken up by a hydrogen, which isn't that helpful. It's one of the reasons why we like to raise the pH of soil so there are fewer hydrogen ions around, and so therefore more of these negative sites can be taken up with some of the nutrients that we know plants really want. All right, so just to indicate the different charge states that different minerals can have, different clays can have, compared to organic matter. All right, so we've got they're on the left-hand side, and then on the right-hand side, we've got the relative ability to hold positive charge. If you can hold positive charge, that means you're that negative. So this is also a table about how much negative charge these things have inherent and built into them. All right? So notice, let's start from the bottom. Sands, not a lot of surface area, no charge embedded in them as they form. There's a charge balance for them embedded as they form. No excessive negative charges. No ability to hold anything. Sands are just rocks that are broken into small bits. Silt, same story. Sands broken into small bits. But with clays are not silts that are broken up into small bits. That's not how you get a clay. The way you get a clay is you dissolve a rock in the soil water, and then that soil water dries out, And as it dries out, you precipitate something holy and brand new. You precipitate a clay particle. And as that happens, these negative charges get built in. And if you want to know the details, you have to look at Dr. Weil's book. We could talk about it, but we don't have time. The iron oxide clays, they don't have a lot of negative charge, not as much. They tend to have more positive charge than the other clays, right? But then you go up the list and you start to get these different types of clay formed in different environments, starting with different soil water with different concentrations of silicon, aluminum, and everything else. And you generate these clays that just have more negative charge embedded in them, therefore they can hold more positive charge. But of course, the star of the show when it comes to being able to have negative charge that can grab positives is, look at that organic matter, right? That organic matter is just great stuff. So just like Emily was saying, you know, you've got the dirt that you've got, but the way that you manage it to introduce organic matter can change things. You might have a really sandy soil. Well, if you can find a way to add organic matter to it, you're going to have something that instead of holding no nutrients can hold a certain amount. And instead of something that holds no water can hold a lot more water, right? You can also have a material that starts off very clayey, right? Sticky when it gets wet. Roots are going to rot in place because as that soil gets wet, there's going to be basically no oxygen available after a few days. So the roots won't be able to breathe, they'll just die in place. But add organic matter to that. You've got the porosity, right? You've got more oxygen, the roots can persist. And so you've got the ability for air and water to be exchanged. And you know what? You're adding even more ability to retain those mineral nutrients. So it's never a bad thing. It's never a bad. You have to go to a real special case to find a situation where you have too much organic matter. All right. Should you rake your leaves into the street? No. Okay. That organic matter has been hard fought by your tree. Right, for 80 years, 100 years. You know, it took it 120 years to get that big to grab the nutrients that it did that year. Don't rake those nutrients into the street. Don't rake that organic matter into the street. Keep those nutrients, keep that organic matter. All right? Do not be like your neighbors. Keep that stuff. And I challenge you, you do an experiment. Have one little spot where you rake the leaves and the dirt's bare. Have another spot next to it where you put your leaves. You wait a year, right? And you move what remains of those leaves after a year, and you will see a huge difference in the structure of the soil. Not necessarily the color, because it's only been in 1 year, but you'll see a huge difference in the structure of the soil, all right? Organic matter is this precious thing for so many reasons, just like Emily was telling you. All right, so, very good. Some soils are better at holding nutrients than others. Sometimes they have no more nutrients because that's just the way they were built, or it's the way they were managed because we got more organic matter in there in particular. All right. So this relative ability of soils to be able to hold charge has a name. Okay. It's called the ion exchange capacity. Ion exchange capacity. And of course, there's two types of ions, right? We have positively charged ions. If you remember from chemistry class in high school, what's the name for a positively charged ion? A cation. Nice. So therefore, we have something called cation exchange capacity. And then we've got this other thing, you know, you're remembering from high school, this table is prompting you, right? The negatively charged ions are called anions. And so there's also this anion exchange capacity. And it's measured in the amount of charge that can be retained by a certain amount of soil. All right? If I've got 1 kilogram of dry dirt, how many positive charges can it retain? That's the cation exchange capacity. If I've got 1 kilogram of dried dirt, how many negative charges can it retain? That's the anion exchange capacity. And I know that a lot of people say that soil's not dirt, so don't use the word dirt. I'm using dirt in a loving term. All right? So it's got a name. These things have names. They're a thing. Here are the units associated with them. All right? It's the amount of charge that you can hold per mass of soil. So what are the units going to be? It's going to be an amount of charge per kilogram of soil. And funny unit for the amount of charge, all right? Centimoles of charge. You're just going to have to deal with that, all right? You're just going to have to deal with it. Centimoles of charge. It's not on here. It is from high school. What is a mole?
It's a specific number, but I can't remember what it
It's
is.
a specific number. Think of a mole as simply being a big dozen. How much is a dozen? 12, all right? So we can say 12 or we can say dozen, and we mean exactly the same thing, all right? Dozen works great when you're talking about things like egg cartons, okay? That's why we have that concept. It's an incredibly useful concept when we're talking about eggs. Is it a useful thing when we talk about grains of rice? No. There's no recipe book that says take a dozen grains of rice or 700 dozen grains of rice, right? Are there any? There's no recipes that are like that, all right? So it becomes a useless unit at some point. The same thing, and merely the same thing, is true of a mole, all right? A mole has a utility in some contexts and no utility in others. The context where a mole has utility, and remember, a mole is just like a big dozen, it has utility when we're talking about numbers of atoms, all right? If we're talking about numbers of atoms or numbers of molecules, that's when it has utility. And we say that a mole is like a big dozen because a mole is simply a huge number. And it's got to be a huge number because atoms are so darn small that if we're going to talk about working with them in kind of a meaningful way, like in the lab where I'm going to say, I'm going to pick out from, my jar of chemical, so many fractions of a mole of stuff. Well, it turns out that moles just work really well, right? If you have a handful of carbon atoms, you've got about a mole of carbon atoms, all right? And a usable amount that we can work with. And so the detail here is that a mole is 6 times 10 to the 23rd of atoms usually, or molecules. So that's basically a 6 with 23 zeros after it, or 6 with 23 zeros after it. That's a really big number. And it has to be big because we're talking about working on a human scale with something that is so small. All right? So don't be intimidated. You can be intimidated by moles, but I encourage you not to be. They're just a big dozen. Dozens have a place, right? Moles have a place. Is there a mole of eggs on the planet? No, that number is so big there are not even a mole of eggs on the planet. All right. If I give you a dozen atoms, have I given you anything that you can deal with? No, right? So it just doesn't work. Right? But if I give you a mole of atoms, that's a handful, literally, and you can work with that. All right. So that's what we mean by moles. So it turns out that a kilogram of soil doesn't contain a mole of negative charges, for example. It contains about 1/100th of a mole of charge. And so that's where we get the centimole from, right? 1/100th of a mole. We're using the metric system. There's no kilogram of dirt out there or probably no kilogram of dirt out there that has a mole of negative charges, right? So that's why we don't use that unit. we'll use centimoles because most soils out there, you can measure the amount of charge that they have in 1 kilogram in terms of hundreds of a mole. Still a very large number. Now there's another unit that's exactly the same thing. If you switch from one unit to the other, nothing changes to the number we're talking about. It's called milliequivalents of charge per 100 grams still of soil, okay? They're the same thing. So on your exam, they're gonna use one of those units. And does it matter? It does not matter, okay? They mean the same thing. So just, you got this, okay? You got this. All right. All soils, just like we said before, have a distribution of sand, silt, and clay. They also have a distribution of different types of clay minerals, all right? So soils are going to have a distribution of different cation, their ability to hold cations, okay? Sands, well, if it's a pure sand, a truly pure sand, it's going to be really small, like 0. But very rarely is there such a thing as a pure sand. Usually there's a little bit of clay in there too, some clays that are stuck to the sand particles, okay? So we're actually going to get some cation exchange capacity, even in something that we classify as a sand. Remember Emily's textural triangle? If you're going to be a pure sand, a truly pure sand, where are you in that texture triangle? Not just in the bottom left region, you're at the very bottom left corner, in the corner, because that's where you have 100% sand and nothing else, all right? No soil exists in the corner, right? It's usually displaced a little bit inside of the corner because there's some sand and clay there, all right? Okay? But obviously with clays, we're going to tend to have the most negative charges, right, that are there, and so therefore the highest cation exchange capacity. All right, and so centimoles of positive charge per kilogram of soil, or milliequivalents of positive charge per 100 grams of soil, same number. Sorry about that, but it's simpler than you think. You don't have to, there's no conversion to do. Okay, so let's do a cation exchange question, and I'd be surprised if there isn't a question like this on your test, okay? So we went to the lab, we had a sample of soil, and we extracted from that soil all of the different ways that positive charge can show up in the soil, okay? We had in 1 kilogram of soil, we had 3 centimoles of positive charge that was there because of hydrogens that were stuck to the soil. We have 12 centimoles of positive charge because of calcium that was stuck to the soil, and on down. And we want to know, what's the cation exchange capacity of the soil? All right, what are the units associated with cation exchange capacity? Centimoles of what? Of positive charge, that's right. It's not centimoles of atoms, it's centimoles of positive charge per kilogram of soil. Now here's the thing, what are you getting? Are you being told centimoles of atoms, or are you being told centimoles of positive charge? You're being told centimoles of positive charge. This ain't hard. Yeah. The numerator is centimoles of positive charge. The denominator is kilograms. We already know we got this from 1 kilogram. So we're going to add those numbers up. Each of those numbers are centimoles of positive charge. We're going to add them up, and we're going to have our numerator, and there it is, okay? 25 centimoles of positive charge per kilogram of soil. We got it just by adding it up. Can you do this? Yes, you can do this, okay?
Is it not wanting to go?
No, we're just enjoying this moment. I'm enjoying my banana bread. Hug your banana today, right? Now here's what this looks like in real life, okay? This is a scanning, tunneling, electromicroscopic graph picture of a clay particle in real life. And you can see how beautifully how this is. In real life, atoms actually show up as calcium 2+. This is not a real picture. This is a diagram, all right? But this is how it works, okay? Now remember what the clay particle is. The clay particle is not broken up rock. It's actually a crystal that is formed out of the soil water, all right? And so these kind of sheets here that you see that have this elegant geometry, these represent the crystalline sheets of a clay particle, all right? And we know that clay particles have embedded negative charge in them, that it's going to be attracting these positive minerals, right? And so here's what that looks like. If we could take a picture of a clay particle, right, we'd see the crystals, and then if we could somehow label the atoms that were there, it would kind of show up like this. All right? Here they are. They are stuck to the clay particle because the clay particle has these negative embedded charges on it, right? And here's something that's so interesting. They got stuck. Are they permanently stuck? No. They can be moved. What if all of a sudden you made this environment much more acidic and you added a bunch of hydrogen atoms? What could they do? Those hydrogen atoms are going to kind of be seeing from around the corner. They're going to be seeing the negative charges that are embedded here. And they might whack this calcium off, right? So now we have a calcium in solution. You could add some kind of iron atom, and it could whack the calcium off. And now we have the calcium in solution. So these things, they can desorb, all right? Hence this idea that we say cation exchange capacity. Cation holding capacity is what clays have, but because they can hold the clays, they can also let them go. We give it the name cation exchange capacity. Cation hold-on capacity is the same as cation exchange capacity. Cation exchange capacity is a real name. So this is a very interesting idea. The chemistry of what's in the water is related to the chemistry of what's stuck on your clays. All right? They're exchangeable. If you change the chemistry of the soil water, it will change what's stuck on the clays, right? And so these things are constantly interacting. Whatever's in the soil water is exchanging with what's on the clays, and they interact. But this is one of the mechanisms that supplies cations to the soil water. And how about this? When roots take up nutrients, where are they drawing the nutrients from? This is very important. Our roots kind of reaching into this clay particle and kind of brushing off a calcium ion and kind of scooping it up. No. Roots are in the soil water, and they are grabbing their nutrients from the soil water. They do not grab them from the surfaces of the soil particles. They just don't do that. Roots will take up what is dissolved in the soil water, so it's got to be dissolved. So this is a way that we hold cations so that eventually, through one mechanism or another, they're going to dissolve into the soil water and a plant can get them. This is a reservoir that in and of itself isn't directly available to plants, but over time is. And so it kind of looks like this, all right? Here they are stuck to the soil, but they're interacting with the soil water. Some are getting stuck to the soil, some are coming off. When they come off, they're dissolved, and then they can be taken up by plants. Nice picture. This is a great instructive picture for you. All right? So in the soil water, that's what's dissolved and that's what's plant available, okay? But they can stick to the soil solids and therefore kind of be in this reservoir for future access, okay? And then hydrogen's special, right? We have a special measure of it called pH. And because hydrogen is a positive charge, if there's a lot of it, it can go, and there's a lot of it dissolved in soil water, it can take up space on the mineral surfaces. So we call active acidity the portion of the hydrogen ions that are dissolved in the soil water. That's what you're measuring in a pH test. You're measuring the active acidity. But then you could have the hydrogens that are absorbed to the cation exchange capacity. They're stuck to the soil minerals. We call that the reserve acidity. So this is all, think of it as kind of one system that's kind of interacting. They're not separate, right? They're interacting.
Excuse me, I just want to make sure I'm more hydrogen in soil water lower the pH?
Say it louder, ma'am.
The higher the amount of hydrogen in the soil water, the lower the pH?
That's correct. That's the funny way that pH works. And you just got to get used to it. There's a couple things we have to apologize for in kind of soil fertility, and one of them we have to apologize for. We just got to get used to it. Is if you've got more hydrogens, you've got a lower pH. If you've got more hydrogens, you have more acidity. All right, so low pH means more acid. Low pH means more acid. And if it's helpful to write that down, go ahead and do it. Low pH means more acid. There needs to be a rap song about that so that our young people in the United States can kind of, it'll be helpful. All right. Okay, so ion exchange capacity. Now here's the interesting thing. The relative sizes of these things, it looks like the relative sizes are about the same, but it's actually not true. The amount of the amount of charge that exists on the soil because of clays and organic matter is much, much bigger than the amount of charge that's going to be dissolved in the soil water at any one time. All right? And like we said before, acid conditions means there's more hydrogens are around, and so there's going to be more hydrogens stored in the soil solids, and therefore they're taking up a space that a nutrient could occupy. And so that's called pH-dependent cation exchange capacity. You lose cation exchange capacity if conditions are more acid, right? Because you've got these hydrogen ions that are taking up space where you might want a calcium or a magnesium in that place. A lot to that slide. Take the cations away, and this is what the clay particles really look like, right? These clays are formed with these negative charges embedded in them. This is what it actually looks like. This is the cation exchange capacity. These aren't cations, these are the negative charges that allow the cation exchange capacity to exist, right? This is what the cations are interested in. Those charges are built in this way through this process called isomorphic substitution. I am sorry, I think you actually do need to know that name for this test. So this idea that negative charges are built into clays as they form through this process called isomorphic substitution, I think you need to know that. If you want the details, you can look in Dr. Wild's book. I don't want to spend too much time on this. The idea here is that in a coffee pot, you've got an indicator, right? A little indicator tube, and you've got a reservoir. The relative sizes are very different. The reservoir is like the amount of cation exchange capacity that the soil solids have, much bigger. The indicator tube is like the amount of dissolved charge that's present in the soil water. But actually, if we were really going to make it more the way it really is, we'd make that reservoir so much bigger. Dwight, tell me about lunchtime.
Well,
Should we take a break?
Do you still have a fair amount to do on this presentation yet? Like 10, 15 minutes? Could you stop right now?
We can stop right now.
If you don't, pardon?
So if you
Yes.
Yeah, let's stop. Let's stop right
Okay,
now.
stop.
Seems like it's halfway.
Okay, why don't we go ahead and stop right now, because we're probably disrupting the meeting back here in the back anyhow. So why don't we go ahead
The
and.
food is hot.
Yep.
Let's do it.
Let's take a minute.
You guys are